let's now look at the action potential, and for the action potential to actually happen, there are two things happening. One is depolarization and the other is repolarization. Now, before that, when we had discussed about resting membrane potential, that was the polarized state. So now we have to move to the depolarized state. Now, if you remember the scenario, inside there were potassium ions, outside there were sodium ions, correct, in the extracellular fluid. Now, please concentrate on the diagram that comes up here for your better understanding. Now, whenever a threshold stimulus is applied, what will happen? The beginning of the nerve impulse will take place. Now, what is that? Because of the excitation, the voltage gated channels are opening. Now, you can see there are two channels over here. One is a sodium channel and one is a potassium channel. Now, I had told you that at one particular time, only one type of ions can enter or exit. So here you can see that as the threshold stimulus is applied, the sodium ion channels have opened and there is a rapid flow of the sodium ions from outside. Alright, what they are showing here is nothing but the interiors of the axon and outside it. Okay, the extracellular fluid where the axon is placed. Then the movement of the sodium ions inside is happening at a very fast rate. Now, because of that, what is happening is the positivity inside will increase. Earlier, the scenario was interiors were negative. Now, not in the first instance, but throughout the axon length, this will happen one after the other. Alright, and because of the rising of the positivity or the positive electrical signal, the next adjacent sodium gates are opened. Alright, it is because of the change in the electronegativity, correct? So, the changes in the electrical difference is going to initiate the opening of the further sodium gates, which are the voltage gated ion gates, correct? Now, as the sodium keeps on entering inside, there is a shift of the charges. Inside it will become positive and outside it becomes negative, and that is the depolarized state. Alright, so let us write down. When the threshold stimulus is applied, the voltage gated sodium channels, sodium ion channels open and there is an inflow, alright, influx of sodium ions from the extracellular fluid, okay, to the inside. Now understand, the membrane here is of utmost importance because whatever changes are happening, it is happening at the length of the membrane, and so the difference across the membrane is responsible for this change, and it happens here because the ions are densely populated in this particular path, alright, inside the axons as well as in the extracellular fluid, but they are very, very dense here. The ions are very, very dense here, and the way they have depicted the positive and the negative sign shows that the accumulation of the ions are just very close to the membrane, very touching the membrane or adjacent to the membrane. Then, now the rapid flow of the sodium ions inside will then, what it will happen, there is a difference in the local electric potential. All right. So, to the inside, as a result, there is a local electrical potential difference, and you can see that it goes like a wave. All right, the positive charge inside keeps occurring faster throughout the membrane, so that brings about the local electrical potential difference, and the negativity is wiped out, or the electronegativity is wiped off. Then the interiors inside, it has turned now positive, and outside it is now negative. Now because of this, and now the wave of action potential has passed through, the changes has happened. Inside it has become positive, outside it has become negative. That is going to initiate the opposite process, because the depolarization has occurred, the action potential has done its work. Now the neuron has to come back to its normal state. All right, this is the way it will reach till the axon endings, and then whatever changes will happen there, that we will see later, but this is how the changes has happened. The action potential is created, which is the action potential is created, which is how much? 55, and it is positive, 55 millivolts. You remember there was negative charge inside, that was minus 70, and now the inside has turned positive, and that is the action potential that can be measured. It is positive, so plus 55 millivolts. So this is how the action potential is now moving further, and it keeps happening in the adjacent parts of the axon till it reaches to the place where it has to reach to. So that is depolarization. Following depolarization, what will happen is exactly the opposite, that is repolarization. Now here the process will be exactly opposite, but how it is brought about? Because of the increased positive atmosphere inside, the sodium gates will now close and the potassium gates will open, and now the potassium ions will start moving, and that will bring the neuron, the activated neuron, back to its rest, and that is repolarization, exactly opposite of depolarization. Now between these two, there is a process. Now this entire process, it takes place, the entire process takes place between, don't be surprised, between 1 to 5 milliseconds. All right, and so we don't even realize what is happening. The nerve impulse goes, comes back, and the action is actually brought about. So the entire process takes place, entire process of depolarization, action potential and repolarization will take place between 1 to 5 milliseconds. And then comes, after this comes a refractory period. All right, so what is the refractory period? Refractory period just after a nerve impulse has passed through, no stimulus will be able to excite a neuron. All right, it's kind of taking rest before the next impulse comes, and that period is refractory period. It is about 1/1000th of a second in a large myelinated nerve fiber. All right, of a second in a large myelinated nerve fiber. All right, what it is? The inactivity of a neuron in spite of being stimulated by a stimulus. All right, so that is the refractory period. And then there is something called as saltatory conduction here. Saltatory conduction. Now this word is taken from a Latin word of saltare. So saltare means to hop or to jump, and this has shown that the nerve impulse is faster in myelinated nerve fiber. Now what happens is the myelin sheath actually insulates the entire axon, but there are certain constrictions where the myelin sheath is coming closer and almost not there, which is at the nodes of Ranvier. Now at that nodes of Ranvier, there is no myelin sheath, and so the signal's action potential will jump from one node of Ranvier to the next adjacent node of Ranvier, and that is how it fastens up or quickens up the process of the nerve impulse, and that is the saltatory conduction. So what is saltatory conduction? The jumping of the nerve impulse from one node of Ranvier to the next. All right, and can you imagine the speed it can achieve because of this myelinated nerve fiber? Up to 120 meters per second. That is the speed taken up in the myelinated nerve fibers. We can mention it here. It goes as much as up to 120 meters per second. All right, and in case of the even when you talk in terms of the spending of energy by a neuron, the myelinated nerve fibers are much economical because of this saltatory conduction, hopping conduction, and less amount of energy is wasted. Whereas in the non-myelinated nerve fiber, what will happen? The action potential has to go throughout the length of the axon, whatever would be the length, and that much of exchange of this sodium and potassium through that exchange pumps, and that will involve so many ATP molecules. So that is less economical, and this is more economical because of the saltatory conduction. All right, and what is the word taken? Saltare, or it means to hop or it means to jump. Then now, next we move on to see the velocity of a neuron. Now, on what does the velocity of a neuron depends? Let's write the velocity of a neuron depends on one, whether it is myelinated or non-myelinated nerve fiber, and second is the thickness of the diameter of a nerve fiber. All right. And what is it? What is the observation? Thicker is the nerve fiber, faster is the nerve impulse. The nerve impulse will be conducted. So we can write it here. Thicker fibers means faster conduction of nerve impulses. The velocity is faster in case of a thicker nerve fiber as compared to a thin nerve fiber. And of course, the myelin sheath or the non-myelinated condition of the nerve fiber also matters a lot, and we have seen the reason why, because of the saltatory conduction. Then now we move on to the exchange of the chemicals that is happening in case of the transmission of nerve impulse. Now we come across a new term here which is called as synapse or synapse. However you want to pronounce, you can say that your spelling stands synapse. Alright, synapse or synapse. Now what is a synapse? It is the junction between or the place where the end of an axon meets to the other end of a dendron. Alright, so there is a gap. There is no direct connection between the cytoplasm of the axon and the adjacent dendron. No, there is a proper gap. Now to understand this, just a very simple example which I am sure you all must have seen. If you ever observe the two bogies that are attached in a rail, alright, the railway compartments or two bogies of a train are attached. Now they are definitely attached with a cord, but you must have seen that extremely high magnetic endings are there. You know, there are those parts which are almost touching each other, but there is a distinct gap between the two, and that is because of the high magnetic field that is created there, and that is the way the nerve endings are also there. They almost come closer, but they do not really touch each other. Alright, now look at the diagram provided here. Now that is what you can see here is actually the end of an axon, which appears like a bulbous structure. Alright, so we will see in details what exactly makes up a synapse or a synapse. Let's define synapse. The junction between the end of one axon and the end of a dendron. Alright, between one axon ending and the another dendron ending is the place that is called as the synapse. Now, the terminal endings of the axon will not have the myelin sheath, and they will be protruding out like bulbs, all right, swollen structures that you can see here, and that is called as the synaptic knob. All right, it's also called as synaptic button, or we call it as synaptic knob. Now, inside that knob, what you can see are circular sac-like structures, all right, and inside that sac, there are the chemicals which are going to carry the impulses, the neurotransmitters, okay. So, there are oval or round sac-like vesicles called as the synaptic vesicles, which are filled with chemicals, the neurotransmitters. All right, the neurotransmitters. Now, we had seen that in the sympathetic nervous system, there was noradrenaline, adrenaline, and in parasympathetic, it was acetylcholine. So, these are the different types of neurotransmitters. In fact, there are many others, but these are the main ones that we usually come across. So, these are the neurotransmitters that are there inside these vesicles and which are called as the synaptic vesicles. And they are the ones which are going to actually carry the impulse further to the next adjacent neuron. Now, the gap between the two, as you can see, there's one synaptic knob there and there's one synaptic knob down. So, between the two, you can see the gap, which is the synaptic cleft. Here, let's mention inside the knob, inside the synaptic knob. All right. So, synaptic knob is the swollen tip. All right. The swollen tip is the synaptic knob, and inside that there are synaptic vesicles. Now, synaptic cleft will be the gap between the two synaptic knobs is called as the synaptic cleft. Okay. Now, when we are seeing that there are two neurons involved, obviously for a nerve impulse to happen. So, what we will name them, the first one, in the one which is going to send the nerve impulse, will be the presynaptic cell, which will also have the presynaptic membrane, because the membranes are involved, correct? So, presynaptic membrane. And which is this? It is the neuron which is going to send the nerve impulse. All right. And then comes the receiver, which is called as the postsynaptic. And the terms are very simple for you to remember. The above one which is going to send, and the post is the one which is going to receive. So, postsynaptic cell and the postsynaptic membrane. It is the neuron which is receiving the signal, which is receiving the signal, all right, or receiving the nerve impulse. Okay, so that is about the post synaptic cell or post synaptic membrane. Now, before we move further as to how this synapse occurs, we have to understand that there are two types of synapse: electrical synapse and there are chemical synapse. So we will see the difference between the two, between the electrical synapse and the chemical synapse. But before that, let us understand what is the how is the this vesicles, the synaptic vesicles, they will be having the neurotransmitters, but they need a carrier, and this carrier is brought about by calcium ions. All right, so we will see exactly how the process happens here right in front of you. Let's look at the mechanism here in front of you. What you can see is that now the moment a nerve impulse arrives at the axon ending, that is at the synaptic knob, there it will initiate the movement of calcium ions. All right, now from the cleft, these calcium ions, the gates open again, the membrane gates open again, and they enter, the calcium ions enter inside the synaptic knob. After entering, what they are doing is they are actually attaching themselves to the vesicles there, the synaptic vesicles, and they are carrying them right up to the membrane. But once they reach the membrane, by the process of exocytosis, the vesicles are releasing the chemicals. The neurotransmitters which are loaded inside the vesicles are being released at the cleft, and from there the chemicals will be going across to the other side of the cleft and other side of the synapse, that is the post-synaptic cell. So from the presynaptic, it has come with the help of calcium ions. They are actually binding to the vesicles, individual vesicles, and bringing them to the membrane. Now at the post-synaptic cell, what is happening is, you can see that there are receptors which will receive the neurochemicals, the chemicals that are sent by the presynaptic cells, and then they are sent to the dendrons, and from there they are carried further. So this is how the exchange happens, and it depends on which type of nerve fiber is involved, and accordingly the neurotransmitters change. There are as many as between 24 to 30 neurotransmitters have been identified, but acetylcholine and the adrenaline, noradrenaline, these are the main ones, and also dopamine and serotonin are also neurotransmitters. There are plenty of them, but for us we need to understand that it is acetylcholine, noradrenaline and adrenaline are working as the main neurotransmitters. Then how will the transmission stop? What will happen? The neurotransmitters are sometimes, then they are absorbed by the knob. The synaptic knob will absorb some of the neurotransmitters and some will just degenerate, and that is how the impulse will then stop, because you cannot continuously stimulate a nerve fiber and it cannot go on happening. All right, it doesn't happen that way. There has to be breaks, there has to be refractory periods, there has to be rest, and this is how it brings about. The knob itself will absorb some of the acetylcholine or the other whatever neurotransmitter is there, and some just gets degenerated, and that is how the process will stop. So you saw the moving of the chemicals, the neurotransmitters with the help of calcium ions, they are moving right up to the membrane, then they are released by exocytosis, taken over by the postsynaptic cell, and further it moves. So that is how the mechanism of a synapse actually happens. Now what we need to understand is that there are two types of synapse that occurs in the human body, and let's see what are they. There is electrical synapse and there is chemical synapse. Alright, now what is the difference between them? The electrical synapse is brought about by the electrical difference, the potential electrical difference which brings about which is transmitted across, and that brings about the nerve impulse. So first point is it is brought about by the potential electrical difference, all right, and that is between the two nerve endings. Here it is brought about by chemicals which work as neurotransmitters. Alright, so that is first point of difference. Second point of difference is that the synaptic knob here is not clearly seen, whereas in the chemical synapse we just saw that the synaptic knob was very, very clear. So the synaptic knob is not very distinct. Here it is distinct. Here it is definitely much faster and here it is slower. Alright, and if you look at the cleft size of the electrical synapse and chemical synapse, it is way apart. Here the synaptic cleft is of 0.2 nm and here it is 20 nm. It is that much is the difference, almost 100 times bigger than the electrical synapse. So it is 20 nm in chemical synapse, okay, in chemical synapse. Then the last point that we can see is that here it is less common. It is not very commonly seen in case of the human body. It is less common and here it is more common. Alright, so that brings an end to the difference between the electrical and the chemical synapse, and now we need to proceed further to the disorders seen in the nervous system.